When you're working with RNA, you have to take precautions to make sure that that RNA does not go away thanks to RNases like RNaseA. And so we can do things like clean surfaces with RNaseZAP, use nuclease-free water, potentially DEPC treated water, and doing our reaction, we can add RNase inhibitors that are little proteins that are going to bind to these RNAs, so these RNA chewers, to prevent them from chewing up our RNA. And then we can do things like wear gloves, use filter tips, so these tips have little filters in them on the pipette tip so that you're not like shooting stuff into your sample. And we have to do all of this because RNases, so these RNA chewers, are like everywhere, and so we have them on our skin, they're in the dust, they're everywhere because we like secrete them, and other like microbes secrete them and stuff to protect themselves so they can like destroy foreign viruses and stuff before they get to them. But when we're wanting to work with RNA, we need to be really careful that we avoid these RNases, and they're everywhere, and they're really durable, and so we have to take special tricks to work with them. So here's a little bit more about how we do it.
So RNA, ribonucleic acid, it differs from DNA in that it has this second OH group here. So DNA is deoxyribonucleic acid. Deoxy, it doesn't have this OH group, this hydroxyl group. And there's also a U instead of a T. But the bigger thing here for us in terms of working with RNA versus DNA is this 2' OH, because it's going to make the RNA both inherently less stable as well as more vulnerable to attack, because this OH, it can kind of get like bored and attack this phosphate group. So this is what we call a nucleophile. This, especially when it gets deprotonated, so it has this proton removed. This oxygen, basically, it is going to be nucleophilic. It has more electron density than it wants, much more on this in other posts, but it is going to be attracted to this phosphorus group, because although this phosphate group might be negative overall, these oxygens, because they're electronegative, they're pulling away the electrons from the phosphorus, making this phosphorus partly positive, so this O can attack, and then you get this chain breakage. You can have this happen just like under basic or alkaline conditions when this will get deprotonated. But you can also have it happen with the help of enzymes. So enzymes are reaction meter uppers or speed uppers. And what they can do is they can kind of help promote the deprotonation of this, and then get this oxygen in this vulnerable state and under optimal conditions to attack itself. And so really it's taking advantage. RNases like RNaseA take advantage of this in order to get the RNA to attack itself and cleave itself. And so we need to take precautions in order to both avoid the RNases and to inhibit those that we can't avoid. So we're going to be talking about some of the strategies we can use to clean our workspaces, to avoid introducing more contaminants once we clean things, and to inhibit anything in our reaction. How do we actually get rid of the RNases that are already present? So the RNases that we're caring about, these nucleases, so these ribonucleases, so these RNA chewers. So there are different types of nucleases, and some of them, so for example, there are nucleases that cut DNA as well. The DNA, though, remember, it doesn't have that. It doesn't have this 2' OH, but it can still get cut, but you can't use the O to do it. And so there are some nucleases that use things like metals to help them. And so we can inhibit these by adding a chelator, something like EDTA, which is going to bind to those metals and kind of hide them. So a chelator is like a claw. It's going to bite down on this metal and prevent the nuclease from using it. But this is not going to work for something like RNaseA, because what RNaseA is doing is it's going to actually be getting the RNA to attack itself. And so we can't take away this, because then we wouldn't have RNA, then we'd have DNA, right? And so we're trying to protect our RNA, so we need to prevent this RNase from doing its thing. So this common RNase mechanism is it's going to kind of create a basic environment inside of the pocket. So it's going to hold on to the RNA in this nice environment where it's going to help deprotonate with the help of this histidine. It's going to help deprotonate that hydroxyl group. And now this is going to be a nucleophile, and seeking the electrophile, like we talked about before, it's going to attack the phosphate group, and this is going to cleave the RNA. And this is an enzyme, so what this means is it's a catalyst. It's going to speed up reactions without getting used up in the process. And so it gets reset, and it can do this again and again and again and again. And using this like 2-histidine method, like back forth, so it can like regenerate itself, get this cut. And so we need to inhibit the RNase, and it turns out that it's really hard to inhibit an RNase. So we have these machines called autoclaves, and these autoclaves are basically like high pressure steam cooker type things. And they get really hot and really high pressure, and you would think that they would be able to destroy pretty much anything, but they have a really hard time destroying RNase A. So just to note that RNase A is the name for the bovine version of one RNase. The most similar one in humans is RNase 1. They're both referred to as like pancreatic type because they're secretory RNases made by the pancreas. But the name's kind of misleading because they're also made by most cells, especially like endothelial cells. So the cells lining your body, tubing and compartments. And so we secrete this in our spit, in our sweat, etc., to add a layer of protection against viruses before they can get in. And bacteria and fungi and other tiny life forms also do this, and they're the main source we worry about in the lab. So in terms of the text I'm going to show you on some of these slides, that's from my blog. I have a blog post on this with more details. But so anyway, RNaseA is a beast. So the autoclave doesn't destroy its activity. And this is in part because it has a lot of disulfide bridges. So cysteine, one of the amino acids, one of the protein letters, it can form these cross bridges, these disulfide links, where it can form these covalent bonds within itself that help like certify it. And so this is going to help allow it to, even if the autoclave like makes it unfold a little temporarily, it has all those crosslinks that aren't going to get hurt. And so once it cools off, now it can become active again. And so the autoclave isn't going to be allow us to get rid of it. So how do we go about removing RNases before we get started? So first, in terms of removing them from like the solutions that we're going to be working with, such as the water or the buffers that we're going to be using, so that we're not like exposing our RNA into these liquids that have RNases in them. So there's this chemical called DEPC. And what it does is it modifies NH, SH, and OH groups. So we're talking about amine groups, thiol groups, and hydroxyl groups. So these like nucleophilic groups. And so what they can do is DEPC is going to alkylate them, because it's going to basically, when the group nucleophilically attacks, you're going to get this, it's stuck on. So before we saw with the RNase enzyme that it had that histidine that was going to attack and pull off the proton and that sort of thing. Well, here what it's going to do is it's going to attack this, but then it's going to break this and it's going to get stuck on here. And so it's going to get like permanently stuck. And so this is going to inactivate the enzyme, but it can also do this to other things. And so you're not want to add DEPC to your solution that has your RNA in it, or that has your other proteins in it, because it's going to modify those things as well, because they all have, like you have in your amino acids, you have all these groups that can react. You have groups that can react in various other molecules as well. And so we need to remove the DEPC before we actually put anything that we want to work with in it. And so thankfully this we can remove by autoclaving. So autoclaving does destroy the DEPC. And so it's going to like split into CO2, so carbon dioxide, and ethanol. When you autoclave it, you might realize that it gets kind of this fruity smell. Don't freak out. it is just that there's these chemicals called esters. So it's like carbonyl, so the C double bonded to an O next to an oxygen. These are formed when you have ethanol during that autoclaving. You're generating that ethanol, and that ethanol can then react, and you can get this fruity smelling ester compounds. And so don't worry about that. But what you might need to worry about is that if you have DEPC that doesn't get completely inactivated. And so like Thermo Fisher suggests you use about 0.1% DEPC. If you go lower, you might not be able to, you might not inhibit all of the RNAs, but if you go higher, you might not inactivate all the DEPC. And if you don't inactivate the DEPC, then this can like cause problems, as we had talked about before. Even if you do inactivate it all, sometimes those ester products that you form can inhibit things. This can be a problem with things like in vitro translation reactions if you're trying to do those. Some other specific examples. And so you can typically now, like, you often like buy these little bottles of this super pure water, this nuclease-free water, and like you can get DEPC version or like non-DEPC, but still RNase free, which is only like a couple dollars more, and then you don't have to worry about these things. But you can also do your own DEPC in the lab. And this, when it comes to what you can be adding DEPC to before you autoclave, you can't use it with all buffers. So you can use DEPC treated water before you to make buffers, but you can't, there are some, like you don't want to add DEPC to a buffer that already contains like tris or HEPs or some other amine containing solutions, because as we talked about before, these amine groups can like react with the DEPC, and they can also like kind of like use up all the DEPC, as well as because you're changing the buffer, now you're going to be changing the buffering activity. They're also incompatible with solutions that can't be autoclaved. So if your solution can't be autoclaved, then you don't want to add the DEPC before to it, because then you can't autoclave it to get rid of the DEPC, but you could use the DEPC treated water in order to make it. Another thing is that some places say like don't use MOPS because you can't autoclave MOPS, but then other places say you can autoclave MOPS. It just turns kind of yellowish, but it's still fine. So I guess it's fine. Yeah, don't know. But one of the most important things to keep in mind when you're working with DEPC treated water is that the water is only as clean as you keep it. And since you've now inactivated the DEPC, you need to like ensure that you're not introducing any other RNases. And so you can do this by using sterile procedures, wearing gloves when you're touching everything. And as we'll talk about, wiping down surfaces. When I work with RNA, I use these filter tips. So these pipette tips, they have this little filter in here. And this way, when you're pushing down on the plunger, you're not like shooting out any sort of dirt or grime or anything with potential RNases. So we want to make sure that so we have the solutions clean and we're pipetting them with clean things. So we have like sterile pipettes. Sterile pipette tips. So in addition to being filter tips, they're also sterile. And now we want to clean our surfaces, and then we'll introduce our stuff. And so one thing, there's some commercial products that you can use to like decontaminate surfaces with RNases, as well as like do it yourself versions. And so I found this really helpful site, Pipette Jockey, and they give this protocol for making your own, as well as telling you, they looked deep dived into what was inside of these. This RNaseZAP that's commonly used, it has this sodium dichloroisocyanurate, which when with water will form this hypochlorous acid, so this bleaching reagent. And what this bleaching reagent is going to do is it's going to act like a bleach, and it's going to oxidize things. So it's going to mess up the proteins, mess up that RNase protein. And so it can basically take OH groups and turn them into carboxyl groups, carboxylic acid groups. It can oxidize like ethyls and stuff. You can also have these chlorine groups substituted. So there's a lot of various ways that this bleach can cause damage. There's also a couple of other things in there. So there is like this, so NaOH, so to make the solutions more basic, as well as SDS, sodium didecyl sulfate, which, as we talked about before, is this detergent. And so it's going to, it's a lowish concentration here. So it's probably not mainly to denature the proteins, but instead to kind of just make it so that you're getting rid of any other grime and stuff and making it easier for the bleach to find all of the RNAs to react with. Okay, so now that we have cleaned our surfaces, you can also clean surfaces by like kind of like wiping them off with DEPC water and then autoclaving them. Or I mean like surfaces of like containers and that sort of thing, not like the surface of your bench. Okay, but anyway, so now it's time to do your reaction. And so no matter how many precautions you take, there still could be RNases introduced in your reaction from like the air, as well as from in the stuff that's in the reaction. And so when I'm going to be working with like RNAs that I'm purifying like out of cells and stuff, there are RNases in the cells, there are RNases in the environment when the cells break open. So there's all those RNases already in there, and I need to make sure that they're not going to chew up the RNA that I'm trying to save. So we can imagine that with DEPC, we are kind of just like going after all sorts of things. So we were going after the active side of the RNAs, but we're also going after the active side of all sorts of things, as well as non-active sides of all sorts of things. And so if we want to inhibit just the RNAs, in our reaction, because there's other stuff in our reaction, we can't have something that's just as like generic as DEPC in terms of what it targets. So we can use these more specific inhibitors, and these RNase inhibitors are like these proteins that are actually going to bind to the RNAs really tightly and inhibit it. So the like classic one is this RNase inhibitor that was originally found in like the human placenta, I think, but now it's recombinantly made. So they stick the instructions for making it into like a plasmid, and then they get, in this case, bacteria to make it for you. But this RNase inhibitor, it's going to bind in this like non-competitive inhibitor, I think. So it's going to bind to this compound in either the substrate bound or unbound form. So whether or not the RNase is bound to RNA. So this is going to inhibit RNaseA family, as well as some related ones, but there's also a lot of other RNases out there. And so this like superazine is an RNase inhibitor that's also a recombinant protein that binds non-covalently and inhibits RNases, but so it inhibits RNaseA, but it also inhibits like BC1 and T1. Although I saw that this is like a lower affinity, so it might take more or longer or something to inhibit, but it gives you it inhibits a broader range of the RNases. And these RNA inhibitors, because these are proteins, you're going to be able to remove them when you do like a phenylchloroform extraction. So happy working with RNA. Hope that helped.